This is Mr. Worski again, and this is the second screencast of molecular biology of eukaryotes. Now, this was the last slide that we saw at the first one, is when we talked about changing the hnRNA to mRNA and how the mRNA needed to be escorted to the pores, and that needed to happen in order to change the protein, or change the, to produce proteins. If you don't have that happening, no proteins produced. And oh well. That's one way to control protein synthesis, is by controlling these steps here. And here's a picture of the 5' cap being added, the 3' poly A tail, the RNA splicing, and if this doesn't happen, you don't have the production of a viable protein. So what else? Let's say that mRNA is produced, right? So far we have this transcription happen, mRNA is produced. Now the mRNA is in the cytoplasm. How else can we control the production of proteins? Control mRNA degradation. So in Prokaryotes, mRNA is broken down right away, it's broken down quickly. In Euks, the degradation can vary. The longer the life of mRNA, the more protein that is produced. So the ends help fight degradation, right? So if mRNA is not degraded quickly, then what else can happen? So let's say we transcribed, we have mRNA, it's staying around for a really long time. How else can protein production be controlled? Translational control. And how can you do that? You can control if the ribosomes attach to the mRNA. There are these things called initiation factors that bind the mRNA, the tRNA, and the rRNA all together. You affect the initiation factors, you affect translation, you affect protein synthesis, you affect the expression of the gene and the production of the protein. Initiation factors. All right, so let's say we have everything in place. We have transcription, we have mRNA, it's not being degraded quickly, the initiation factors are good. How else can we affect translation? Well, miRNA and siRNA can bind to the mRNA and they can stop the ribosome from translocation, which stops protein synthesis and it stops the production of the protein. All right, so let's say we have transcription. We have production of mRNA. We have mRNA in the cytoplasm. It's not being degraded quickly. We have the initiation factors working. No miRNA or siRNA. And we have translation. We now have a polypeptide. What else can happen? Well, that polypeptide might need to be modified. If it needs to be modified, it needs to be modified in the endoplasmic reticulum or the Golgi complex. If a modification does not happen, the protein will not be functional. If you don't have amino acids added, if you don't have them cut, if you don't have the polypeptides added together, if you do not have carbohydrates or lipid added to the proper places, it will not be functional because it will not be the right shape. All right, so what happens if something happens to any of these proteins and they're non-functional? What happens if you have a non-functional protein? Well, those non-functional proteins need to be broken down because they're junk, they're trash, and too much trash or junk will build up and kill the cell. So how does the cell break down defective or damaged proteins? Well, enzymes break those things down. We have proteases to do that, right? But there needs to be a tag on those defective or damaged proteins to tell the enzymes to break them down. And ubiquitin is a molecule that tags those proteins so they can be broken down. Now, so too much trash kills cells. Alzheimer's, that happens. Parkinson, that happens. Huntington's disease, that happens. All these things that you have too much trash because too much non-functional proteins, and that will kill the cell. If you kill brain cells, that's bad. They don't reproduce. So we talked about how genes are regulated, how the expression is controlled, right? How protein synthesis or how protein regulation happens. So in prokaryotes talked about genes on the move. We talked about transposons. But in eukaryotes, there are a couple of different ways that genes can move. Right? Just not only transposable elements or transposons, but the first one I want to talk about are antibody coding genes. In the antibody, there are 3 genes that code for the antibody. We have the C gene, which is the constant region. It codes for the constant region. There are only 5 different of those. The V gene, which is the variable region, and you have at least 400 of those. And then you have the J gene, which binds the C gene and the V gene together. Now, before birth, in each cell, 1 C gene, 1 V gene, and 1 J gene will move out of the chromosome and line up next to each other in front of a promoter. And this will be the antibody that B cell produces. This only happens in B cells because those are the cells that produce antibodies. So we have them lined up. We have all the B genes, we have all the J genes, and we have all the C genes lined up in a line. They rearrange themselves so that we have 1 V, 1 J, and 1 C. And that is the antibody that B cell will produce. We have the V gene here, we have the J gene here, and then the C gene down here. The J gene joins both the V and C regions. The second type of genes that I want to talk about that are moving are done through viruses. So viruses, the vDNA incorporates into the cell's DNA. Talked about briefly with herbs, right? So when the vDNA gets in, they produce the viruses, they can kill cells. However, your body can turn around and get rid of the viruses, but you still have the vDNA in your DNA. The problem is when the vDNA inserts into your DNA, that can cause mutations, and then that can cause the gene that it inserted to not be produced. Now, sometimes that can bind to or insert itself into genes that can cause cancer or that helps prevent cancer. If you have mutations in those things, well, then you can get cancer. Look at HPV. So after you fight off the virus, your vDNA is methylated. Now, eukaryotic transposons. There's only one type of eukaryotic transposon. It's called a retrotransposon. It's not an old-fashioned transposon. What retrotransposons do is they're transposons in the DNA. They're copied into mRNA, and they reuse the enzyme reverse transcriptase to make DNA, and that will insert itself into a new spot in your DNA. So you get that? So we have a retrotransposon. It's transcribed into RNA. Reverse transcriptase makes DNA out of it, and then it will insert somewhere else into your DNA. And when it inserts into your DNA, it's bad because it can insert itself into functional genes and cause mutations. So eukaryotic transposons, nothing good can come from these things. Nothing. So that was the 3 examples of genes on the move in eukaryotes. So we just want to talk a little bit about genes, viruses, and cancer. So not a very pleasant topic, but an important topic nonetheless. So carcinogens cause cancer, mutagens cause mutations, and mutagens are carcinogens, which means the things that cause mutations cause cancer. What is cancer? Cancer, you have cells that divide uncontrollably and invade and destroy normal tissue by preventing the tissue from functioning normally. So they're unspecialized cells, and they hurt the tissue by preventing it from functioning normally. So there are over 200 different types of cancer because each cell type, you have a type of cancer for each type of cell. So the first step is transformation, in which you transform cells to be cancerous. And when you do that, you change the cell's DNA. Now we have a couple of genes. One of them is called oncogenes. And if you change these oncogenes through mutation, the cell will be cancerous. So the normal oncogenes are called proto-oncogenes. These cells usually regulate cell growth, like the RAS gene. Then you have tumor suppressor genes, and they produce proteins that stop uncontrollable growth. So any mutation to these genes, like TP53, can cause cancer. So we have proto-oncogenes. If we have a mutation within the proto-oncogenes, they are now oncogenes, and they will be hyperactive. So RAS, we talked about that before, produces the RAS protein, and that stimulates the production of growth factor receptors. So if the RAS is mutated, it increases the growth factor receptor production. So it binds to more growth factor, which increases cell division. So RAS usually keeps the growth factor receptors in check, but if a mutation to RAS, there's nothing controlling that. Produce a lot of growth factors receptors, they bind to growth factor, and the cell divides uncontrollably. TP53 was a tumor suppressor gene. It's on chromosome 17. It produces P53. Now, P53 is a transcription factor that stimulates the production of P21. P21 binds to the CDK, cyclin-dependent kinase gene, and stops cell division. That's a lot of steps. Now, if there is no P53, if there's a mutation to TP53, you don't produce the P53, you don't produce P21, which does not bind to CDK, and cell division does not stop, so it increases cell division. Now, we know what happened to Henrietta Lacks. We know that the HPV virus inserted itself into the TP53. We know that. And we know that she had cervical cancer because of this mutation, of this insertion. So that's how viruses can actually cause cancer. There are other tumor suppressor genes, BRCA1 and BRCA2. These are the two breast cancer genes. They were discovered in Berkeley, California. That's where the BRCA comes from. Not breast cancer, although it works either way. BRCA stands for Berkeley, California. Now, in colon cancer, which isn't a very pleasant topic, but there needs to be 4 mutations that happen in order for colon cancer to occur. The first mutation is to the APC gene, which is a tumor suppressor, and the cells divide and they form a polyp, which is a growth. The second mutation is to the RAS gene, and then you form an adenoma. Remember, the RAS gene is a proto-oncogene. The third mutation happens to any gene, and that is you form a mutation. And the fourth mutation happens to TP53, and then you form a carcinoma, which is the bad form of cancer. Best way to prevent this is by increasing fiber intake so you keep things moving so your cells aren't exposed to toxins for too long. And that is the end of this screencast. This is the end of eukaryotic molecular genetics. If you have any questions, review the video, read your textbook, talk to me tomorrow. Have a good night.